Welcome to Chemistry Classes by Vijay Kumar Sethi. We were discussing Unit 14 Biomolecules, and in this video we will discuss about proteins, classification and structure of proteins, and denaturation of proteins. First, proteins. Proteins are the most abundant biomolecules of the living system. Cheap sources of proteins are milk, cheese, pulses, peanuts, fish, meat, etc. They occur in every part of the body and form the fundamental basis of structure and functions of life. They are also required for growth and maintenance of body. The word protein is derived from Greek word proteos, which means primary or of prime importance. All proteins are polymers of alpha amino acids, and in the previous video we have discussed about these amino acids. Classification of proteins. Proteins can be classified into two types on the basis of their molecular shape. First one is fibrous proteins. When the polypeptide chain run parallel and are held together by hydrogen and disulfide bonds, then fiber-like structure is formed, and that is known as fibrous proteins. Such proteins are generally insoluble in water. Examples are keratin, present in hair, wool and silk, and myosin, present in muscles. Another type of protein is globular proteins. This structure results when the chains of polypeptides coil around to give a spherical shape. These are usually soluble in water. Examples are insulin and albumins. Generally, there is one question: what are the differences between fibrous proteins and globular proteins? I think after this discussion, you are able to write the answer of this question. Fibrous proteins have fiber-like structure. Globular proteins have a spherical shape. Fibrous proteins are insoluble in water and globular proteins are soluble in water. Examples of fibrous proteins are keratin and myosin. And examples of globular proteins are insulin and albumins. Okay. Now, structure of proteins. Generally, proteins may have four types of structure: primary structure, secondary structure, tertiary structure, and quaternary structure. We will discuss one by one. First, come to primary structure of proteins. Proteins may have one or more polypeptide chains. Specific sequence of amino acids. Remember, specific sequence of amino acids in each polypeptide in a protein is said to be the primary structure of that protein. Okay. Any change in this primary structure, that is the sequence of amino acids, creates a different protein. See one example, like these, the three-letter symbol amino acids are represented and these are in a particular sequence. Now if we change the order, suppose this is replaced or then this is replaced with this one. Okay, they change their position. Then we will get the another type of protein. Okay, this is the specific sequence of amino acid gives primary structure to proteins. Secondary structure of proteins. The secondary structure of proteins refers to the shape in which a long polypeptide chain can exist. They are found to exist in two different types of structure, namely alpha helix and beta-pleated sheet structure. These structures arise due to the regular folding of backbone of polypeptide chain due to hydrogen bonding between CO and NH groups of the peptide bond. Okay. We can say secondary structure is two types: alpha helix and beta pleated sheet structure. Now first come to alpha helix structure. In it, a polypeptide chain forms all possible hydrogen bonds by twisting into a right-handed screw, that is helix. Just see, this is a right-hand screw and this is a left-hand screw. You can see the difference between these two from these diagrams. Okay. By twisting into a right-handed screw with the NH group of each amino acid residue hydrogen bonded to the CO of an adjacent turn of the helix. Now just see the structure. This is alpha helix structure of proteins. Okay, this represent the right-handed screw, that is helix structure. There is hydrogen bond between this CO and this is NH. Okay, you can say this is CO and this is NH. Hydrogen bonds are there. Now, another one is beta-pleated sheet structure. In beta-pleated sheet structure, all peptide chains are stretched out. Okay, stretched out to nearly maximum extension and then laid side by side, which are held together by intermolecular hydrogen bonds. The structure resembles the pleated folds of drapery. Okay, drapery means fabric arranged in loose, graceful folds. You have seen, you can see such type of structure in curtains. Okay, like this. This is drapery, and therefore is known as beta pleated sheet. This is the arrangement of this beta pleated sheet structure of proteins. Okay. Then tertiary structure of proteins. The tertiary structure of proteins represents overall folding of the polypeptide chains. That is further folding of the secondary structure. It gives rise to two major molecular shapes, namely fibrous and globular. The main forces which stabilize the secondary and tertiary structures of proteins are hydrogen bonds, disulfide linkages, van der Waals, and electrostatic forces of attraction. Next one is quaternary structure of proteins. Some of the proteins are composed of two or more polypeptide chains, referred to as subunits. Okay, and the spatial arrangement of these subunits with respect to each other is known as quaternary structure. I think this is clear to you. Primary, secondary, tertiary, and quaternary structure of proteins. Now just see, this is the diagrammatic representation of protein structure. This one is primary structure, sequence of amino acids. Okay, this is secondary structure. Is it clear? Now, if the secondary structure is folded more, then we will get the tertiary structure. And if there are more than one subunits, then the arrangement of these subunits gives the quaternary structure of proteins. Clear? Now you can also see primary, secondary, tertiary, and quaternary structure of hemoglobin. This one is primary structure. This is secondary structure, alpha helix structure. Okay, further folding of this alpha helical structure represent the tertiary structure. Okay, and now in quaternary structure, more than one subunit of polypeptide chain present in the protein. Okay, this is quaternary structure. This blue color represent one polypeptide chain, and this is yellow color represent the another polypeptide chain. Okay, and this part, this represent the heme group, that is containing the iron. Okay. Then denaturation of proteins. Protein found in a biological system with a unique three-dimensional structure and biological activity is called native protein. When a protein in its native form is subjected to physical change like change in temperature or chemical change like change in pH, the hydrogen bonds are disturbed. Due to this, protein loses its biological activity, and this is called denaturation of protein. Clear? During denaturation, secondary and tertiary structures are destroyed, but primary structure remains intact. That is, there is no change in primary structure, but secondary and tertiary structures destroyed. Examples, very common examples: coagulation of egg white on boiling and curdling of milk, that is conversion of milk into curd. These are the examples of denaturation of proteins. Okay, thank you.